Can you turn the light off? Oh. Okay. I have the difficult task to talk after Charles. And without any break, I mean, maybe I'll give up on this talk. Okay, but just in the honor of Tzvi, that we really were the best friend, I have to continue. Okay, so one of the best decisions that I made in my life, I mean scientific life, was to go to Tzvi Zellinger and offer collaboration. And moreover, I asked him to leave the Turkey erythrocytes and to go to the fly. Naively, I thought that I convinced him with good reasons that the fly is excellent for biochemistry. Look at the big eyes. And you can collect billions of heads in a very simple technique. And on top of it, you have the genetic power and the molecular genetic power. So I thought these are good reasons. And Tzvi was convinced very quickly. But today I learned that the reason was different. He just gave up on competing with the other guys. This was my impression. So I don't know. But I still prefer to think that he sees the advantages of the fly. Okay, so what I'll do, I'll just give you some highlights from our collaboration. And, okay, so this will be the outline of my talk. So you see, I'll concentrate on this, the discovery of the biochemistry of fly phototransduction by Tzvi Zellinger. He actually discovered it. And then I'll move to a different topic on the TRPO trip channels. What are they? Why are they important? And then I'll go to the specific trip channel, TRPV1, that Charles mentioned it, about the ones that taste sharp food. So though the TRPV1 channel most likely mediate inflammatory pain owing to its distribution and function, direct evidence from humans is still lacking. And so I'll tell you today about analysis of something remarkable that we found here, of the first identified complete loss of function of the TRPV1 mutant in humans. And I'll tell you, this is still ongoing and have not been published. I'll give you some biochemical data and sensory data about this unique human. Okay, so let's go step by step about the discoveries of Tzvi. So what you see here, actually, you heard today about the G protein coupled receptors, and you heard about the similarity between the beta-arrestin and the visual photopigment. Not arrestin, the beta-adrenergic receptor, sorry, and the visual pigment. So in the fly, this I found, by the way, in my PhD, there is something different than the rhodopsin in our eyes. In the fly, I found in the barnacle that the fly is a bistable pigment. It has a dark stable rhodopsin that in the chromophore is in the 11th cis form. And if you shine, in the case of the fly, you shine blue light, then you convert it to a second stable state in the old trans configuration, and this is the stable meta-rhodopsin. And this is photoreversible and photoconvertible. You can illuminate by orange light the M state and give it back. Okay. So this was known at the time that Tzvi entered the field. Another fact was known that the meta-rhodopsin is phosphorylated. This was found first in the vertebrate and also in the beta-adrenergic receptor, with multiple phosphorylation. So this was also known. Another thing that was known at the time, that since this is dark stable and exists for hours, it must be inactivated because we want to see in the light and not to see in the dark. So the lifetime of M we found is only 100 milliseconds. And the reason is that it binds to arrestin. It's bind to arrestin. You heard a lot about this. And it inactivates, prevents the binding of the G. Okay, so this was also known. The arrestin, by the way, there are two arrestin in the fly, a small one, a big one, and Charles. Here. Oh, here. He cloned one of them. The other one was cloned by Hiro Matsumoto. And so this was also known. Okay, so Tzvi, as you know, he is very methodical and he wants to see everything on the gel. I mean, if you don't see something on the gel, maybe it doesn't exist. So he wanted to see the phosphorylated meta-rhodopsin on the gel. And so he used the P32 to label it, and he couldn't see the meta-rhodopsin on the gel. And he was puzzled by this because, I mean, there are 100 million molecules of rhodopsin in a single photoreceptor cell. And it has multiple phosphorylation. So how come you cannot see it on the gel, on the predicted molecular weight of about 30 kilodalton? But Tzvi never gave up, never. And so he changed the conditions. And then he found that if you use calcium-free solution, then suddenly you see a tremendous phosphorylation of the rhodopsin. So this was immediately clear to him that the phosphorylation of rhodopsin is controlled by a very efficient phosphatase, calcium-dependent phosphatase, that immediately dephosphorylates the rhodopsin, the phosphorylated rhodopsin. So this was an important discovery. And the second thing is about the arrestin. So we already knew that there is arrestin. But the arrestin of the fly is phosphorylated. Unlike the beta-arrestin, the arrestin of the fly, as shown by Charles and by Matsumoto, is phosphorylated. So Tzvi could see it on the gel before he could see the rhodopsin. But then he found something remarkable, that if you illuminate the complex of phosphorylated rhodopsin with arrestin, then you cause dissociation of the arrestin after the red illumination. Dissociation of the arrestin, and it goes to the cytosol. And he could measure it. And this dissociation allows the phosphatase to dephosphorylate rhodopsin. And this is how he got to these cycles that you see here. We were sure that this is the correct cycle because in parallel, we all the time competed with Charles, as he told you. And he found it by a different way. Tzvi used pure biochemistry. And Charles used genetics, molecular genetics, electrophysiology. And we came to the same conclusion about the same time, that this is a valid cycle. Okay, so this was one important contribution of Tzvi. The other one was the identification of the visual GQ of the photoreceptor cell. From pharmacology on other species like the horseshoe crab and squid, we already suspected that G protein is involved in the process. By the way, this was one of the arguments, I forgot to say, that I convinced Tzvi to move to the fly. I said there is a G there. But this was all that was known. And again, Tzvi wanted to see the G, the visual G. And so how you show that there is light-activated G protein? So he used what Safi mentioned yesterday. He used photoaffinity labeling. He used not the sophisticated ones that Safi synthesized, but a common azido-anhydride GTP. And he could show that, if you look here carefully, you see that there is a GTP binding protein here of about 38K, but this one is not sensitive to the red-blue illumination. But there is here 41K that you see it only after blue illumination. So this was the first identification of the visual G protein. And also then he showed by binding of GTP gamma S, light activation, blue light activation of this G protein in the Drosophila, also in the housefly, the musca, compared to the red illumination. And now the next question, what is the target of this G protein? So if we follow evolution, Charles mentioned, you are half asleep.
No? I was looking at the slides over there.
Ah, sorry. Okay. The importance of evolution. And I agree with him. But here we have a case that evolution didn't work. Because at that time we already knew from the work on the vertebrates that the target of the G, the visual G, was cyclic GMP phosphodiesterase. And Zvi was also a great believer in evolution. He thought, okay, it must be that also the target of the fly G is cyclic GMP phosphodiesterase. And there were some publications, again from the horseshoe crab, that at that time was dominating the field of invertebrate phototransduction. And so people use pharmacology and they could excite the limulus, the horseshoe crab photoreceptors, by adding 8-bromocyclic GMP or cyclic GMP. So people thought, okay, evolution works. But then this was not good enough for Zvi. So he set a system where he dissected the eye, incubated them in tritiated inositol, and then measured light activation. At that time, he told me, well, I heard something about the phospholipid effect. And I said, what the hell is a phospholipid effect? He said, well, production of IP3 and all this. So since he couldn't see any production of cyclic GMP or something similar, he was looking for the phospholipid effect for production of IP3, IP2, and IP with the DOX columns. And so this was the first demonstration of light-activated phospholipase C, because he, in this retina where he used tritiated inositol, he could show that by blue illumination you produce, in this case, IP2, but also he could show the IP3 and IP. There was a very efficient phosphatase that converted the IP3 quickly to IP2, so he inhibited, but here you can see the IP2. And most importantly, this reaction was greatly enhanced by the presence of GTP gamma S. So, and inhibited by GDP beta S. So this was actually the first demonstration of that in the fly, it's not cyclic GMP, but the inositol lipid signaling. And this was confirmed later on, a year later, by the work of Bill Pack, who isolated a PLC mutant, and he showed, well, this was unknown, like Charles showed it. It's a receptor potential was 0. It was not responsive to light. And then he found that this encodes for phospholipase C. So it was clear that phospholipase C is essential for the vision of the fly. Okay, and so all this, this work, coupling of photoexcited rhodopsin to inositol phospholipid hydrolysis in fly photoreceptor is really a landmark publication, because this was the first time that anyone claims such a claim. And he summarized, all these findings. This was a picture I took from a PhD thesis of one of his students. So you see the components of the phototransduction. This is the rhodopsin, isomerized by photons, activate a heterotrimeric G protein, the GQ alpha bound to GTP, activate phospholipase C, that hydrolyzes PIP2 into diacylglycerol and inositol trisphosphate. Okay, so how do you know? This was all shown in biochemistry. how do you know that this is a unique cascade? And it's essential for vision, because we know that there are more than one G protein there, and there are other possibilities. And here comes the work of Charles. I mean, in parallel with all this biochemical work, they were isolating mutants, Drosophila mutants. Some of them Bill Pax, some of the Charles. He made a mutant of the G proteins, the GQ alpha, G beta, and he made a mutant of the clone and sequence. And clone and sequence the PKC. And so it was clear, and these are the names of the mutations. And it was clear that without these proteins, the fly is blind. Okay, so it was clear that all these proteins that Zvi isolated biochemically, everything was confirmed by, or done in parallel, by isolation of the mutant. This is the power of the Drosophila. But the major question was how activation of phospholipase C lead to the light response. So obviously it activates the light sensitive channels, but it was not clear what are the light sensitive channels. And these are, and I'm going to talk now about them. So I can speak about the work with Zvi, I mean, more than 2 hours, I mean, because we have how many publications? 27, he said. But now I switch gear and go to the main topic of the promised presentation. And this is about the target of this PLC. The target of this PLC. And the other name is, what is the identity of the light sensitive channels? And now I have to go back about 10 years to the time that I came as a postdoc to the lab of Bill Pack. At that time, there were 3 labs in the world that adopted the genetic dissection approach. I mean, using Drosophila to dissect complex biological processes. The lab of the late Seymour Benzer, he wanted to dissect learning and memory. The lab of Martin Heisenberg in Tübingen, Germany, he wanted to dissect visually guided behavior. And the lab of Bill Pack, he wanted to dissect phototransduction. He was the most modest, I would say. And I happened to come, without knowing what he's doing actually, to be a postdoc. At that time, they didn't know much about the systems. And they were collecting mutants. And every postdoc who came to the lab had to choose one of the mutants and to characterize it. And to the great disappointment of Bill Pack, I didn't choose any one of his mutants. And I chose a mutant that was a spontaneous mutation that was isolated in Scotland, and it has a very interesting phenotype. If you keep the fly in dim light or after darkness, then it behaves like wild type. But if you raise the ambient temperature, illumination, then it becomes blind for 1 minute, and then it recovers. So I was intrigued by this phenotype of the mutant. And I said to Pack, well, I want to work on this mutant. And he was very disappointed. And rightly so. I mean, he said, how can you choose a mutant that is based on a single allele that was found in Scotland with no background? And the evidence is not only behavior, but also the electroretinogram that Charles showed you. And at that time, the component of the electroretinogram were not clear. There are 20 different kinds of cells in the retina. So it was not clear if this is a phototransduction mutant. And in retrospect, I think he was correct. But I didn't listen to reason at that time, and I decided to work on this anyhow. And so in order to show that the phenotype really resulted from the photoreceptor cells, I did intracellular recording from a single photoreceptor cell, which was quite challenging because the diameter is only 3 microns of these cells. So this is intracellular recording. This is the intracellular recording from the photoreceptor cell of wild type. And you see the sustained response, intense light. And this is the receptor potential of the mutant in response to increasing intensities of light. By the way, in the invertebrate, the response to light is depolarization. Unlike in our case, it's hyperpolarization. So in response to dim light, this is in log scale, there is a sustained response. But if you increase the light intensity, in log scale here, then the receptor potential declines to baseline during illumination. This is the light pulse. So because of the transient nature of the receptor potential, I decided to call it the transient receptor potential mutant, or in short TRP, or as the Americans call him, the TRIP. And later on it became the official name of the entire superfamily, which is the second largest in all channels, superfamilies. So the main task in the next 20 years was to find what is wrong with this mutant. The people in Scotland, they claim that this mutant has a problem with the recovery of the photopigment. So I checked the recovery of the photopigment, was perfect. And then when I met Zvi, one of the first experiments, I said, okay, let's measure GTPAs in this mutant and see maybe the G is not working. No, the G was working. And I did many quantitative shot noise analysis. And my conclusion was that in this mutant, one of the possibilities is that this is extreme adaptation, because as you can see here also, when you give bright light, the response declined because of adaptation. So one can think that maybe this is extreme adaptation. And so what I found that this is the opposite of adaptation. I call it the reduction in excitation efficiency, because the intense light becomes during light similar to dim light and then to darkness. So I call it reduction in excitation efficiency by measuring the single photon responses. But I had no idea what it can be. And it took a long time. But then, as good things are coming usually by accident, and we heard your talk about the mistakes that you did in the pH. I'll come to this. And so we found something remarkable, that one can mimic the phenotype of wild type into the mutant by adding lanthanum to the medium. So here you see again, we did it in 4 different species of fly. This is in the house fly, but the same thing is in other species. And so this is the control. You see here, dim light, intense light, you see the sustained response. So application of lanthanum. In dim light, it looks like wild type, like the control. But if you increase the light intensity, then you see the transient response. And you can wash the lanthanum slowly, and it recovers. So when I saw this result, it was immediately clear to me what is the function of this protein. It must be a calcium channel or calcium transporter. Must be. And then I look back at all the data, and I noticed that there was something wrong with the calcium homeostasis of this fly. But these were pieces of evidence, not something convincing. And then, luckily at that time, Charles Zucker and independently Roger Hardy, they introduced the patch clamping technique into Drosophila phototransduction. And so since I knew Roger Hardy from a long time when he was doing his postdoc in Tübingen, and I used to work there, I suggested to him to collaborate on this project on the condition that he comes to Israel and teach us the technique and all this. And we did the critical experiments using wholesale patch clamp recording. And I just want to read you the abstract of the Neuron paper that published these results. By the way, we sent it to Nature, and it was rejected on the ground of lack of general interest. So using whole cell recordings from Drosophila photoreceptors, we show that the wild-type response is mediated by at least 2 functionally distinct classes of light-sensitive channels, and that both the TRP mutation and the calcium channel blocker lanthanum selectively abolished one class of channel with a high calcium permeability, and we conclude that the recently sequenced TRP protein represents a class of light-sensitive channel required for inositol-mediated calcium entry, and suggests that this process is necessary for maintained excitation during intense illumination. This abstract stands the test of time, because even today I sign on every word in this abstract. Okay, now what about this recently sequenced TRP channel? It turns out that 2 years before, in '89, 2 years before we published it, Craig Montel and Gerald Rubin, where Charles also did his postdoc, they cloned and sequenced the TRP gene. And they found that it's a transmembrane protein. They suggested that it crossed the membrane 8 times. About the same time, it was independently cloned by Fulton Wong, and he came to similar conclusions. And so they cloned and sequenced, but unfortunately, when they compared to this sequence to the known sequences in the database, it was not similar to anything, to any sequence. And they have a lot of speculation in the discussion, by the way, but they never tried to show that any of them is valid for 3 years. And okay, so when we came to the conclusion that this is probably the light-sensitive channel, it was not clear if this is the channel itself or required for the channel, like in the cyclic GMP channels, that was similar problem. And then Charles, he isolated, among other many mutants, he isolated the gene, clone and sequence the gene of the remaining channel. The remaining channel that gives a transient response, by the way, was cloned and sequenced independently in Australia by Len Kelly, who was looking for calmodulin binding proteins, and TRP or TRP, they are all calmodulin binding proteins, and he found one, and to his disappointment, it turned out to be similar to the already cloned Drosophila channel. So he called it the TRP-like or TRP-like channel. And so we, independently by electrophysiology, decided that there are 2 channels because of the reversal potential and details. And we suggest that this is the other channel. And Charles made a mutant of this channel. And this was very important because he also made the double mutant, the TRP-like double mutant, and this eliminated completely the light response. So it was clear that these 2 channels are required for generating the light response, and there is no need for any other channel. Later on, Craig Montel found another TRP channel that he called TRP gamma, and he claimed that this channel had to interact with the TRPL in order to make the light response. Unfortunately, no one could repeat these experiments, and now we know that this TRP gamma is not expressed in the eye at all. In any case, I mean, the cloning and sequencing of the TRP channel was very important because even at the beginning they didn't know what it is, but it was very important because now that people, for 20 years, nobody cared about this system. And unlike the beta-adrenergic system, I really envy when I heard so many people were interested and working in parallel and competing, and we were alone, and nobody was interested in this, in this problem. It took so long, 20 years. And but suddenly when people realized that we have a new channel, calcium permeable channel, that is the target of the inositolipid signaling. And people were searching for years for such channels because of activation of T cells, because of the store-operated channels. And there was no candidate. And suddenly there was a candidate. So people started to look for mammalian homologs. And after 3 years, the first homolog was found by Craig Montel and independently by Lutz Birnbauer, that was mentioned here. And they called him the TRPC1, and then others were found. They all called the TRPC. The C stands for classical, because they all were found because of homology to the Drosophila channel. Today there are many studies on all of them are very important. This one is the Pheromone Receptor. There is an interesting story on each one of them, and unfortunately I have no time to even to mention how important they are. And then people start to find another member of this family, the TRPM subfamily. The similarity is in the transmembrane domain. I'll show you later on how the structural features of this channel. So the TRPM, the M stands for the first one, the TRPM1, that was found as a tumor suppressor, and now we know that it has also physiological function. This is the channel that in the road bipolar that sign inverting signal in our retina that change the hyperpolarizing to depolarizing response. I'm not going to go to it. And this one is the cold receptor. Again, there is a story, interesting story on each one of them. The TRPM you already heard from Charles. This is the channel of the taste receptors that is activated by all these G protein coupled receptor with the GQ. Okay, so this one is important. And there is another subfamily, the TRPN. Here also, Charles found his family. He found the NOMPC, which is a mechanosensitive receptor. And this is a small subfamily. There is also homolog in the zebrafish. And there is the TRPML subfamily. They were found because of a lysosomal storage disorder in human. And the TRPP, they were found because of a polycystic kidney disease. And the TRPA has only one member, and this is a very interesting channel. I'll mention it today, later on. It's not clear what is the physiological activator of this channel, but it's very interesting. The A comes from ankyrin repeats. I'll show you the structure later on. And it is activated, I mean, there is debate on this, but activated by noxious cold, by many chemicals, and so on. And from now on, I'm going to concentrate on this subfamily, the TRPV subfamily. The V stands for vanilloids, because the first one, the TRPV1, was found by David Julius. He was screening a library, a dorsal root ganglion library, with capsaicin, the hot ingredient of chili pepper, for a reason, because it was known that capsaicin somehow is involved in burning pain. And the reporter was an increase in cellular calcium. And this is how he found the TRPV1, and then he found the TRPV2, and they found all these subfamily. And what characterizes them is that they're all sensitive. They have a very high Q10. They are sensitive to a gradient of temperature, warm temperature. I mean, the cold temperature is TRPM8. And today I'm going to concentrate on the TRPV1. And again, there are interesting stories on each one of them. But the most investigated TRP channel is the TRPV1. I think there are hundreds, maybe thousands of papers on this channel. And the reason is that people think that this is kind of a receptor for pain, yeah, because it's activated by of burning pain. And moreover, after developing a specific antagonist, it it caused in humans, because the pharmaceutical companies are interested in this. They spend more than $3 billion on this in order to find a new kind of pain reliever. And they develop a specific antagonist. And one of the side effects was there was increase in body temperature. And also, these people that took the antagonist, they could burn their hand by putting on hot plate without feeling. So this was discontinued because of these side effects. But they are still looking for. So, as I said, there are hundreds, maybe thousands of papers on TRPV1 because of this. But there are no evidence on human. I mean, it's not entirely clear what this molecule is doing. I mean, we know it is activated by capsaicin, and there is a knockout mouse. And in the mouse, it shows it abolished a phenomenon that is known as allodynia, that a non-painful stimulus becomes painful. Like when you have a scratch, if you put warm water, you don't feel it, but after you have a scratch and you have inflammation, then you start to feel the pain. This is called allodynia. And so this is abolished in this knockout mouse. But there was not a striking phenotype in these mice. So it was not clear. So now my story starts. This was all introduction. How much time I have? I almost go to the... So I'll do it quickly, because this is not... Okay, so the TRPV1, just for introduction, I mean, it's expressed on the pain pathway, and the antrodromic stimulation of this pathway releases many inflammagens, but it's initiated also by tissue injury, and this is the inflammatory soup. So it is both a effector, like in the Drosophila. At the beginning, when they found that it is activated by heat and by pH and by capsaicin, they call it receptor, because they thought like the ionotropic receptor. It activates directly. But then later on, they found that actually it is also similar to the Drosophila channel. That is effector. It is activated by a cascade, as I told you. So we know that this is both a receptor and effector at both times. And it's a multimodal channel, and it is, people think that he is doing almost everything. Okay, so where my story starts? It starts when I gave a talk in the Faculty of Medicine about TRP channels, and then a pediatric neurologist came to me and told me something amazing, that a boy came with his family to his clinic in Mount Scopus, and this boy can eat hot chili pepper like you eat apples without feeling of anything. So obviously we were very intrigued by this, and so we tested. The people who did the work are sitting here. I'll mention them later on. So we tested his sensitivity to capsaicin. And as you can see here, I mean, this is a control, mostly the student in the lab. Here you see we increase the capsaicin concentration, and you see the sensation of hot, in brackets. This boy doesn't feel anything, even if you go to very high concentration. Then this is of capsaicin. Then we wanted to, there was a question here about what is taste. So we because this was on the tongue, we obviously wanted to see if his taste perception is normal or not. And it's absolutely normal. We tasted for salt, sour, bitter, and sweet, and this control and the human subject, and there was no difference. And then we wanted to see if this insensitivity is confined only to the tongue or to the whole body. And so, as you know, if you put and capsaicin or mustard oil, then you get a local edema, local inflammation. That one of the characteristics is that there is a rise in temperature. So we check the local temperature with the infrared camera. The circle here is the point of application. So this is the control. I mean, mainly the people in the lab, but not only. Also, we checked for the children, and this is the human subject. And as you can see, in the control, there is increase in temperature of this circle. In the boy, actually, it was cooled down. And we interpret this as that the capsaicin was dissolved in ethanol. So we think that this reduction in temperature reflects the evaporation of the ethanol. So with all this information, we ask, what is the reason for the incidence of hot chili pepper? And a most probable reason for this phenotype is abnormal function of TRPV1, the capsaicin receptor. And so we perform exon sequencing of TRPV1 locus, which revealed a homozygote single base change in the anchoring repeat domain. If you look at the structural features of all TRP channels, they have the general structure of voltage-gated channels, mostly potassium channel, because they are tetramer like the potassium channel. They have the 6 transmembrane domain. Usually in potassium channel, this is a voltage sensor because of lysine and arginine. This is the pore region, this is S6. And here, this is the anchoring repeat domain that exists in the TRP. Now, unlike voltage-gated channels, they don't have the anchoring repeat. And the S4 residues, I mean the lysine and arginine, in the TRP channel, all of them, is replaced by non-polar residues. And we know that this is not voltage activated at all. So we look, then we decided to do also a whole exome sequencing in order to be sure that the phenotypes come from this single mutation, not from something else. This is a missense mutation. It's one amino acid change. And you can see it better here. I mean, these are the families are all that we checked are heterozygotes, except for the single homozygotes. Okay, so now with all this information, we want to know what is wrong with what the mutation is doing to the channel. Okay, so to do it systematically, we make stable lines of hex cells that can be activated by tetracycline. So we could control the level of expression. You see the different level of expression. We choose 2 clones that were similar in the wild type and in the mutant. And then we could control the expression level simply by the time and the concentration of tetracycline. This was very useful for a quantitative study. And as you can see here, we tested the function by using calcium imaging, because as you know, the TRP channels are calcium permeable, including the TRPV1. So as you can see, when we applied capsaicin, 1 micromolar, there was increase in cellular calcium. This is in different cells, this is the average. But remarkably, in the mutant, there was no effect of capsaicin. Even if we increase the concentration of capsaicin to 100 micromolar, and even if we increase the expression level 10-fold, there was absolutely no response. So we tried all the known activators of TRPV1. We tried capsaicin, we tried resinoferrotoxin, which is a very potent activator or agonist of TRPV1. We tried low pH. We tried the tarantula toxin, was published recently by David Julius, This is the most powerful activator of TRPV1. In fact, he used it when they solved the structure, atomic structure of TRPV1 to compare the open from the closed state to the open state. They used this toxin because with capsaicin they couldn't see the difference between closed and open state. And of course we tried heat. With the heat we got a small increase, but this was unspecific because we got it in untransfected cell as well. Okay, so we also tried electrophysiology because this is more sensitive than imaging. And again, by application of capsaicin, we could get in the wild type, we could get the typical IV curve of the TRPV1. And this is before and after putting capsaicin and wash, it comes back. Here you can see at a specific voltage, the current, it's as a function of time. You see the large increase in wild type in the current. In the mutant, in the patient, in the mutant channel actually, yeah, in the X cells, there was absolutely no effect. I want to go back here as an important positive control. We had no response. In order to show that everything is kosher here in our experiment, we put ionomycin, which is a calcium ionophore. And as you can see, when you put ionomycin, immediately you see the expected rise in cellular calcium. Okay, so here again we see no effect, absolutely no effect. And this is a summary. This is in capsaicin, but we repeat it in pH and other agents. And so our conclusion from all these experiments was that the TRPV1 does not respond to the common TRPV1 activator. So what are the possible reasons for this lack of inability to activate the channel? The TRPV1 mutation interferes with the gating of the channel, rendering it inactive. This, by the way, is an interesting possibility because there are ways to inactivate the channels, but they are all confined, and there are mutations, they are all confined to the pore region. But this one is far away in the anchor and repeat. So it's an interesting question, how a mutation there. A mutation, by the way, that is conserved along evolution. This amino acid is conserved in all the TRPV and all the TRPC subfamilies. So it must be something important there. But it was a novel mutation, unknown. And so, but there are other possibilities that the TRPV1 mutant does not reach the plasma membrane, or that it does not assemble properly to form the tetramer. So we had to check all these possibilities. And so this is a biochemistry that summarizes all these possibilities. So here we used surface biotinylation, and we got the same expression as the plasma membrane in the wild type and in the mutant channel. We also try to see if it assembled properly. So we express the TRPV1 wild type in the mutant and in the presence of TRP conjugated to GFP to see if they form a tetramer. We immunoprecipitated with GFP antibodies, and we got the same result in the wild type and in the mutant. So it seems that it assembled properly. Also, we wanted to check by using semi-native gel if it forms tetramers. And again, we could see by using mild conditions, we could see the tetramer both in the wild type and in the mutant. And then if we make harsher condition, we could see the dimer, and even harsher condition, we could see the monomer. So in conclusion, the mutant channel reached the plasma membrane in a similar manner to wild type. The mutant channel assembled properly, and hence the mutation interfered with the gating mechanism, rendering it inactive. Okay, so we also, all these experiments were done on tissue culture cells. And people may say, okay, maybe in tissue culture cells, something is not like in the native system. So we made tremendous effort to find a similar, expression of the mutant channel from the tissue of the boy. But we have the Helsinki Agreement and everything. This is a boy. And you cannot just take and do such experiments. So luckily, we found that in his blood cells, in what is known as a PBMC, the peripheral blood mononuclear cells, if you isolate this tissue that includes mainly B cells and KT cells, then we could show that in these cells there is the messenger RNA in the mutant and in the heterozygote, the family of the boy, and in the control people. Yeah? We see expression of the TRPV1 in these cells, which is interesting by itself, because we could change this PBMC into B lymphocyte, to immortal B lymphocyte. It's called lymphoblastoid, and there we saw the same picture. And on the Western blot, also the PBMC of the wild type, the heterozygote, and the homozygote. Here in the Western, there is a reduction of the TRPV1 relative to the controls, but we found variability also in different wild types, different people of the lab, by the way. There was a variability between this and this. So we, at the moment, because we have only one sample, this is what we have. But we have the lymphoblastoid, and then there we see a better result. Okay, so also one reason for the insensitivity could be like in peripheral neuropathy, that there is a reduction in the nociceptors in the skin, and we did one biopsy on the boy, and we went to expert on these biopsies, and she said that these skins look normal. We believe her. Okay, so the last slide is what is the most important thing, that we did quantitative sensory testing on the boy, on the family, and on control people, which are the people from the lab and their kids. So what we did here, we use the pain clinic in Hadassah, and they have a calibrated probe that you put on the hand, and you can give a temperature ramp or at different speeds, and then the subject has to report, I feel the heat, don't feel the heat, feel noxious heat, noxious cold, and things like this. So these are experiments that we did. So we go slowly on this, what you see here. So we try it in a control condition, and after we make a local inflammation below the probe. And the inflammation was done either with capsaicin or with mustard oil, which is AITC. It's the activator of TRPA1 that usually is known to be expressed in the dorsal root ganglion together with TRPV1. And recently there was a paper about noxious heat sensation in mice in Nature, and they show that in order to eliminate noxious heat in the mice, you need to knock out 3 different TRPs, the TRPV1, the TRPA1, and another one, TRPM3. Okay, so we tried the activator of TRPA1 and the activator of TRPV1. So the black is before application of the production of the neurogenic inflammation, and this is after. So in the wild type or heterozygote, as you can see, this is before, this is capsaicin. After we put the capsaicin and we cause neurogenic inflammation, there is a change in the noxious heat threshold. You see, it goes to lower temperature, and this is a phenomenon of allodynia that I told you later. This is also in the heterozygote. In the in the boy, in the homozygote, there was no effect of capsaicin, which fits with what we saw on the tongue and what we learn about the property of this molecule. Okay. With mustard oil, this was more difficult because it turned out that this boy was extremely sensitive to mustard oil. Usually you put it at least 5 minutes. With him, you put 1 minute and he couldn't bear it. It was painful for him. So actually, this was, I would consider it as unsuccessful experiment because for the control people, it was too short to do anything. For the boy, it caused inflammation, but it was too short to see anything. So this is not revealing anything. What we also tried to do is to find his heat tolerance, meaning you put the probe, you heat it to high temperature, and tell him to say when the pain is unbearable. So this is heat tolerance. So for the control people, it was in this degree, about 46 degrees. And here we found something different or unique to this homozygote, the mutant, that his heat tolerance was pretty high, 50 degrees, which is quite hot. He could bear it. Another interesting thing is about noxious cold. So again, we checked for noxious cold. This is in the control. In the presence of capsaicin, the noxious cold temperature went down, and this is in the heterozygote. In the human subject, I mean the boy, his sensitivity to cold was very high. You see, at elevated temperature, already he felt cold, and there was no dispersion at all. I mean, this was a clear effect of cold temperature. He was very sensitive to cold temperature, and then we talked to his parents, and they said that he feels all the time cold, even in the hot summer of Israel. I mean, he always has to wear warm clothes. So this is something abnormal that we still have to investigate. So this is one, and this is the other. And of course, there is a difference between the control and the capsaicin. Here, there is no difference, as expected, because he is absolutely not sensitive to capsaicin. With the mustard oil, we got the same thing. It was insensitive to the difference between control and mustard oil. But again, it was highly elevated without dispersion. Then we tested also noxious mechanical stimulus by putting calibrated needle. And again, in the controls, you can see the before and after. You see that there is what is called mechanical allodynia. They became more sensitive after you put the capsaicin. With the mustard oil, there was no result. But with the boy, even the short time of mustard oil show tremendous mechanical allodynia. In summary, this is the first identified complete loss of function in human TRPV1 mutation. Application of the non-activate of TRPV1 resulted in a robust TRPV1 wild-type activation, but had no effect on the expressed mutant. Biochemical analysis revealed proper tetrametric assembly and plasma membrane localization of TRPV1 mutant. The patient has no apparent health problem, except for this extreme cold sensitivity. Shows normal sensitivity to the 4 taste modalities, but total insensitivity to capsaicin. Quantitative sensory testing revealed abnormally high heat tolerance and abnormal sensitivity to noxious cold compared to control groups. The patient revealed mechanical allodynia under TRPA1 activated inflammatory condition. And this study is expected, it's ongoing, and not been published, to promote our understanding on the participation of TRPV1 in human pain physiology and in inflammatory pain. And generation may help to new TRPV1 agonists with therapeutical value. And actually, there are many people who participate in this work. The driving force and the people who did most of the work that I presented are Ben Katz, who is a lab manager and senior researcher in my lab, and Rachel Zaguri. They did most of the work. Simon Edwardson, he is the MD who brought to our attention this boy. And he, in all experiments with humans, you need MD around to make that everything is kosher. So he fulfilled this job. In addition to Hannah Maayan, also in the same department, Eliad Davidson, he is the head of the pain clinic, and he graciously gave us his laboratory, but he was too busy to go around. But anyhow, I mean, he was very helpful. And Orly El-Peleg, she is the head of the Department of Human Genetics in Hadassah, and she helped us a lot with the whole exome sequencing. And the rest of the people were involved in projects that I didn't have time to tell you. We also did a screen with bacteria that we expressed this mutant and wild-type channel, but I didn't have time to go to this. And finally, I mean, this is a team who really did the work. Ben Katz and Rachel Zaguri, they both. And they were helped by Rita, Tal, and Elisheva. And thank you for your attention.